I have one little announcement I want to make. For that, I share the screen. So I'm very proud to present, to tell you that we will have the first EVOLTREE conference this autumn in September. It's on the topic of genomics and adaptation in forest ecosystem, and it will be held online only or hybrid, we don't know yet, at VSL in Switzerland. If the registration has opened, you can register and submit your abstract since this week. So you can go to the page I mentioned here, or you can visit the EVOLTREE website, evoltree.eu, I think, and there you find more information and also a link to the registration page. So now I hand over to Felix Guggerli, who is moderating again today. And Felix Guggerli is also from WSC Switzerland, and he will introduce Ophélie and he will moderate the discussion after the talk. Felix, the floor is yours.
Thank you, Christian. And I'm not sure if we can call this floor or rather screen here. But anyhow, glad to have you all here on this second seminar. And before introducing our guest today, our speaker, I would like to briefly introduce our network, EVOLTREE, for those who have not been in the series or are not part of the network. So this network of excellence that was formerly supported by the European Commission during 2006 to 2010 is nowadays a European research group made up of 30 European research institutes and universities. And I also see that Antoine Kremer, who initiated all of this, is in the audience today. Great to see you, Antoine. So these research groups are all involved in some way in research on evolutionary biology and forest ecosystems at large, but strongly focused on trees and their associated organisms. And the main activities of EVOLTREE involve primarily science and knowledge exchange, such as this online seminar that you're joining today. EVOLTREE's funding small projects allows for mobility grants or conference visits. And we also have training activities that are open for participants and largely students. So today's second talk of this seminar series is given by Ophélie Ronce. And of course, I was familiar with this name. I have not met Ophélie personally yet, but when looking at Ophélie's website and the literature list, I was quite impressed on the diversity of topics that she covers. So Ophélie is a senior researcher and the head of the Evolution Demography Group at the CNRS Institute of Evolutionary Sciences in Montpellier. The major topics, as I condensed them out of this information that I retrieved, is that she studies how life history traits, demography, gene flow, and ecological niches interact to allow populations to persist, grow, and eventually adapt under climate change. So maybe in short, it's evolutionary and ecological variability of all kinds. So Ophélie applies a broad variety of approaches from empirical to theoretical, using simulations, modeling. She looks at large and small scale, and this is all really nicely reflected in her list of literature and shows that she has published accordingly in a broad range of dedicated journals. Ophélie is also currently the president of the European Society for Evolutionary Biology and acts as the vice president from the non-North American part of the Society for the Study of Evolution. So in even shorter words, I think it's best if I let Ophélie tell us what she's actually doing in more detail, and I look much forward to hearing this talk today. Ophélie, the screen is yours now.
Thank you, Felix. So I will share my screen. Up. So thank you very much for the great honor of speaking to you all. So as maybe Felix didn't mention, it's quite recent that I got interested specifically in trees. And I had the pleasure of working with Antoine and other people in Avignon and with Sally Atkin, more recently in Canada, trying to understand evolutionary challenges associated with trees. And I got interested with them because of their peculiar life history and because they are kind of amplifying different questions about generation time and gene flow that I'm interested in general. So today I will present a talk. So I just want a warning that this is very similar to a talk that I gave actually when I came back from Canada in the Turku meeting at ESEB. So I guess some people in the audience might have already heard that. So they can leave now. I won't be offended. And what I wanted to tell you about is whether trying to integrate thinking about evolution of niche, ecological niche with life history theory could help us understand the consequence of climate change beyond being an interesting conceptual subject in itself. So I also want to acknowledge various sorts of funding and mention that because I'm going to talk about theory mostly, you have nice pictures of my vacation during my sabbatical in Canada to entertain you if you're bored by equations and more conceptual things. So, oh, it's not working. Yeah, it's working. So as you know, species confronted to climate change can persist and adapt either by a combination of tracking the climate to which they were historically adapted to through space by migration, or by changing their niche limits to adapt through phenotypic change to a different climate. And this can be achieved by phenotypic plasticity or by genetic evolution. So this has been discussed many times. And also what is, there's a growing awareness that really the life history of organism is really important to understand how species track in space and in physical space and in phenotypic space this shifting climate because of long generation time or life history traits such as dispersal rates are constraining this ability of tracking in those different dimensions. What is less often discussed is the fact that this process of tracking favorable climate in space and through evolution is also changing the life history of organisms that are confronting those challenges and affecting the evolution of their life history. And this is what I want to illustrate today. Conceptually, in terms of modeling, and I think more generally in the way we think about climate change and those adaptive challenges, often we assume that there are a number of phenotypic traits that affect the population, the contribution of individuals to population mean fitness and population growth, as shown here, and that there's some optimal value of this set of phenotypic traits that maximizes the contribution of individuals to population growth. So there's some kind of optimal value of the phenotype. And we often assume a Gaussian fitness landscape just for mathematical conveniency when we model this kind of challenges. And often the way we also model this and think about this is thinking that this optimal value of the phenotype is changing often linearly across some kind of environmental gradient. And this could be temperature, it could be any other environmental descriptor that we want. So it's nice to simplify reality to try to understand it, but it's also nice not to be a bit prisoner of those assumptions and try to question them and see how much this framework is actually matching what we understand and witness about adaptation to climate change in real populations. So this whole notion of an optimal phenotype is really important when we are discussing the value of phenotypic plasticity in helping species to persist under climate change. So generally what we do when we try to assess whether phenotypic plasticity will help is that we assume that the change in phenotype due to phenotypic plasticity will allow a species to express a phenotype closer to the optimal phenotype, depending on some environmental cues that could be climatic factors. So the question often, if we want to do that, if we want to assess adaptive value of phenotypic plasticity, is to be able to determine where is the optimum. Is this optimal phenotype real first, and where is it? Are we far or are we close to it? So we can address this question empirically by measuring fitness and phenotypes in many different environments and trying to relate both and find out if there's indeed an optimal phenotype and how this optimal phenotype is changing with the environment. So there are increasingly data trying to do that, but it's really hard to measure because measuring fitness in itself is hard to measure, especially if you're working with trees, which are very long-lived and where part of the life cycle, such as fecundity and reproduction or recruitment, are really hard to assess and measure and not typically known for a lot of data that we have. So not necessarily as an alternative, but as a complementary approach, what we propose is to use our understanding of the mechanistic relationships between traits and fitness in different environments to try to predict and not measure directly fitness landscapes in different environments to determine optimal phenotypes and how they would change with climate and climate change. So this was a project of a postdoc, Julie Gauzet, in our group, and we worked on this project with various colleagues from Montpellier and from Bordeaux and Avignon through some project that finished a few years ago. So we implemented this approach, taking as an example two elevation gradients that people in Bordeaux have studied for many years on tree species, oak, beech, and fir, and where they have long-term data about a number of about the performance and the phenology of trees along those elevation gradients. They have good climatic data about those. And we try to understand selection pressure on a particular trait, which is bud burst date, the date at which the buds open in the spring, because those traits have been shown to be really important in the response to climate and also to be determinant of species limit in some instances. So we use as a reference, so for the first study we did on that, we're using climatic data from the 60s to the early 2000s as a reference climate. And what we did to predict what would be the optimal value of the trait in those different environments along those elevation gradients is use the PhenoFit model that was developed by Isabelle Schwinn and colleagues 20 years ago now and developed continuously since then. So this model was initially used to predict species range limits for trees across continental scales. And what the model used as an input is a daily climate and a set of species-specific parameter set that determine how the timing of major events in the life cycle, such as phenological traits, such as bud burst date, flowering date, fruit maturation date, depend on the sequence of daily climate data across the whole year. And those phenological traits and timing determine, in turn, resistance rate of the plant to frost or drought, for instance. And together, those resistance trait and phenological traits determine the reproductive success of an average tree for a given year of climatic data for a given species. And so all the parameter sets are from experimental data, they're not from distribution. And the model is producing two outputs for every location and year in climate, which is the date of events, the phenological traits, the date of bud burst, the date of flowering, and the probability that a tree will successfully produce a crop of seeds that year. So if you repeat this process across many years, you can compute the average reproductive success of a tree given its average date of bud burst during that period. And so if you repeat that, so what we did is that normally those parameter sets are fit for every species. And what we did is just a perturbation of this model, changing some parameters in the response of phenology to temperature to generate different dates of bud burst for a given year, and look at how it would change the average bud burst date across the period and change the average reproductive success. So if you relate the two, you obtain this kind of fitness landscapes showing the average reproductive success of trees in different locations. So the different colors are a prediction for different elevation along those gradients in the Pyrenees as a function of the average bud burst date of the tree in that period. And so this is done for beech, oak, and fir. And you can draw a number of conclusions from those predictions about those fitness landscapes. You can see that in all cases, but more strikingly so for deciduous species, the maximal fitness, even at the optimal value of bud burst, is declining with elevation. In particular for oak, you can see that at high elevation, you predict no reproductive success, whatever the value of the phenology, suggesting that those very high elevation population will be actually demographic sink, persisting only by the immigration of seeds from low elevation populations. And what you can also see is that the shape of those fitness landscape is changing with elevation, with not only the maximum, the value of the phenology maximizing fitness being different, but also those landscapes are narrower when you increase the elevation, meaning that if you are in more constrained climates, you cannot you don't have a lot of degree of freedom in terms of the kind of phenologies that allow you to reproduce successfully. And typically, those fitness landscape do not have a Gaussian shape. They have obvious asymmetries. They have also this kind of flat tops, suggesting there's a window of optimal phenology rather than just one value maximizing fitness. So this was for the historical period, and that was published in a paper in Evolution Letters last year. But now we're doing that for, I will show you that later. But so again, for historical climate, what you can see is you can now compare what trees are doing by looking at the plasticity of trees. So those are the red cross. That's the prediction of the model about if you look at their reaction on to temperature, what trees would do if they were planted at different elevation. And the black dots are what is the optimal date of bud burst at those different elevations. And the gray area is showing range of bud burst dates that allow maintaining a fitness that is not less than 5% smaller than the optimal fitness you could have in this environment. And so for both beech and fir, the plasticity allows you to stay pretty close to the optimal date of bud burst, suggesting that indeed phenotypic plasticity here is adaptive. But at least prediction of our model are predicting that for oak, plasticity is not adaptive or not so adaptive. And in particular, the data of bud burst seems to be much later than what is optimal to do in those different environments. And you have a higher sensitivity to temperature or in plasticity that would be optimal, which is a pattern that is called hyperplasticity in the literature and is pretty hard to explain, but I won't have time to tell you about that today, but we have also new interesting prediction about why this might happen, and this involves associative mating. So what I was trying to tell you is that we've started to look at that also in future climate, not in current climate or historical climate, and look at how those fitness landscapes are changed by climate warming. So this is just one example for beech trees. You have here the fitness landscapes for historical climate, and for two scenarios of future warming, one moderate, one larger warming. And an interesting feature, at least for this gradient, so it might not be a general finding, is that in this case, what we predict is that climate warming is increasing fitness at high elevation without, for that species, decreasing fitness at low elevation. And it seems that it actually relaxes selection on but birth date everywhere, rather than really change the position of the optimum or change the, or on the contrary, select for a narrow range of phenotypic values. So I guess because on those gradients, precipitation is not really limiting for at least beech trees, what we find is a signal of relaxed selection rather than more intense selection in future climates. This is not true for oak, but this is kind of true for as well. But those are preliminary results. Don't do much about that. It's just to tease you a little bit. So the conclusion of this first part was that adaptive plasticity, plasticity seems to be adaptive in fir and beech. We find our modeling suggests hyperplasticity in oak, and also suggests that evolution of bud burst date is unlikely to change high elevation niche limits in that species than in others, because as little options to improve fitness due to evolution of that trait, suggesting that other traits might be important for at high elevation in terms of adaptive response. And an interesting conclusion from that it suggests is that along those environmental gradients, other things are changing in those fitness landscape than the position of the optimum, as theory has mainly focused on, and that this change in the widths of the fitness landscape might be something important to consider in our predictions. I just wanted to mention that this study somehow converged with some of the conclusion that we had previously obtained by using the same kind of approach, but at a much broader scale, at the scale of whole Europe for different species as well, where we had this kind of thought experiment. We used this Phenotypic model to predict what would happen if trees were not plastic and if they had the same bud burst date every year and everywhere, which of course is not true, but it's just helping to see actually as a control what is the effect of plasticity on fitness. And our conclusion, so this was for future climates as well, and our conclusion was that indeed phenotypic plasticity, so the red area here is showing places where in future climates plasticity is helping the species to persist and increase its fitness. And the blue area are areas where, conversely, if trees were not plastic, the species would have a higher fitness in future climate without plasticity. And interestingly, it really depends on the species, and it depends where in the range you are. And so plasticity is helping a lot, except at the cold margin of oak trees, and except for Scots pine, because here the plasticity, the sensitivity to climate, because of the requirement for cold temperature in winter to break dormancy of the bud, is actually more adaptive in a warming climate. And this is something that people know, but it gives a very clear prediction of what it does. So at least those studies suggest that phenotypic plasticity might not be sufficient to adapt to a warming climate, at least in some part of the range or for some species, which has for consequences that we should expect genetic evolution to favor changes in those reaction norms as the climate warms in those conditions to track the changing climate. So historically, already in the early 90s, a number of quantitative genetics models were developed to try to understand the kind of adaptive challenges a species facing a changing climate or have to overcome. And so in this early model by Lynch and colleague, what they were assuming that through time was some optimal phenotype changing linearly through time. And initially, the mean phenotype was matching this optimal phenotype. As the optimal phenotype is changing, the mean phenotype already at the beginning does not evolve very fast because it's not very maladapted. And so there's a increasing lag between the optimal phenotype and the mean phenotype in the population. And as this lag increases, the selection to change the mean phenotype in the population are increasing because the selection gradient becomes higher and higher. And the population starts to evolve faster and faster until it evolves exactly as fast as the optimal phenotype is changing through time. And so it's evolving as fast as the climate changes, but it's always tracking the optimal phenotype with a lag, with a constant lag. And if this lag here is too large, so if the mean phenotype lags too much away from the optimal phenotype, the population growth rate might go beyond 0, which is the case here in this example, and the population might go extinct. Not because it does not adapt or does not adapt fast enough, but because it's lagging too much behind the optimal phenotype. And ultimately, this big lag is due to the fact that initially it was evolving too slow to catch on the changing environment. So this is very interesting, and a lot of people have tried to use this model, try to understand what was the critical rate of climate change that would cause the population extinction, and you could try to measure the parameters in this model, try to predict which species might be doomed by climate change or not. One complication is that more and more data about different species suggesting that there's not a single fitness landscape, but there are actually different phenotypic traits maximizing different life history components in the life cycle. So for instance, in this plant, they found that the flowering date that maximizes the survival of the plant is very different. So here in blue, is very different from the flowering date that maximizes the fecundity of the plant. So inspired by this kind of examples, we asked the following questions. So the first question is, how do those conflicting selection pressures across different fitness components affect those lags in adaptation that you predict in a simple population? Second question is, in return, how do those lags in adaptation affect the life cycle and the life history of the plants in a changing environment? And are there feedback loops between those two components that might affect the dynamics of adaptation to climate change? So to answer this question, we built a model, and this project involved Olivier Coteau, Linnea Sandel, and Luis Miguel Chevin as collaborators, and was published in a special issue about maladaptation in AMNAT in 2019. So we had a very simple, we have general prediction that are true for any stage structured population with any number of stages and any kind of complexity in life cycle, but I will illustrate those predictions with a simple 2 stages life cycle where you have immature plants that can survive and stay mature. Once they've grown enough, they mature and become reproducing adults, and those reproducing adults can survive and stay in the productive stage, and they produce immature plants through production. And so similarly to the example in Bacteria stricta, we assume that the evolving trait, which could be flowering date, affects the adult survival and their success of reproduction. So, but with similarly to Bacteria stricta, we assume that the value of the phenotype maximizing survival and maximizing fecundity are different, and both are changing through time because of climate change. And for simplification, we assume that they change at the same rate, or they could change at different rate as well. So let me just introduce you to some quantity that demographers use a lot, and that are really also important to understand what's going on in those examples where the population is made of individuals that are heterogeneous in their demographic properties and not making the same contribution to population growth. So there's a quantity called demographic elasticities that some of you may have come across, which actually measure the effect of a perturbation in a transition in the life cycle on the exponential growth rate of the population. And those quantities are used in demography to understand the effect of disturbances affecting some stages or some transition in the life cycle and others. But they were also used a lot to understand the strengths of selection acting on different life history components. So for instance, those are the quantities that you compute to understand why selection strength is declining with age, and which is the ultimate explanation of the evolution of the aging and senescence in many species. So why am I speaking about demographic elasticities? It's because our model is showing that in a constant environment, if you look at the phenotype that maximizes total fitness, when you've got this compromise between fecundity and survival, because not the same value of traits are maximizing both, you find that this evolutionary compromise is actually an average of the value of the phenotype maximizing fecundity and survival weighted by those elasticities. So the effect of a perturbation of fecundity on population growth rate and the proportional effect of a perturbation in the survival rate on the population growth rate. So if survival is making a big contribution to the population growth rate, the optimal value of the trait will be displaced toward the value maximizing survival, and the reverse will apply if fecundity is making a big contribution to population growth rate. So that is nice. Now, what's happening when the environment is changing, as with climate warming? So in a constant environment, we've got an optimal compromise between survival and fecundity, and we start to change the climate and increase temperature, for instance, favoring earlier flowering time for both fecundity and survival. What we see in our model is we predict an increase in survival through time and a decline in fecundity. So if we were to witness that in a natural population, if we would see that survival increases under climate warming and fecundity decreases, it would be very tempting to think that actually warmer climate are selecting for this shift in life history, that indeed it is advantageous to increase survival under a warmer climate and decrease fecundity in this warmer climate. But in our model, it's not the case. In our model, actually, the optimal value of the compromise between survival and fecundity is still the same as in a constant environment. And the reason why life history traits are changing is different. So what's happening is that through time, so the optimal value of the phenological trait like date of flowering is advancing, as do the optimal value maximizing fecundity and survival. And as for some unstructured population, the mean phenotype in the population is lagging behind this optimal integrative optimum. But because of this lag, because it's not evolving fast enough to actually follow and track very closely the optimal phenotype, the mean phenotype is further away from the value maximizing fecundity, and by accident somehow closer to the value maximizing survival. And this explains the shift in life history that we see, which is actually maladaptive and not the sign of a different life history being selected for in a warmer climate. So this just is interesting because it shows that the fact that you're tracking a changing environment is changing your life history somehow. Another interesting aspect of that is that there are indeed feedback loops affecting the evolutionary dynamics in this case, because what's happening is that if there's some mismatch between the optimal value of a trait maximizing a life history component and the value of the mean trait in the population, this would actually depress this life history component, making, for instance, fecundity lower or survival lower. And as this life history component declines, it means that its contribution to population growth rate will also decline, which means that its demographic elasticity will be smaller, and the strength of selection on this life history component will be smaller. And so because of this initial maladaptation, you weaken selection on this life history component and relatively increase it on the other life history component. So those feedbacks, so this creates more mismatch and so on. So the maladaptation is pushing selection in the same direction as this initial maladaptation. So the selection will start to favor trait values that were initially maladaptive because of those feedbacks. So an interesting consequence of that is that if you look at different scenarios, we have sometime population that would evolve trait values that are favorable for survival. So survival is increasing, but start to invest almost nothing in fecundity. So fecundity is vanishing, and of course, not reproducing is never a good idea for a growing population. So the population actually goes extinct, but the survival of individuals is still increasing while the population is crashing. So this also draws attention and caution about the fact that if we are measuring only one life history component, as we often do in trees, like growth or survival only, we might be missing completely the picture because we are not seeing that the population is actually going down because of lack of fecundity and recruitment. So to conclude on this part, we found that indeed lag in adaptation result in change in life history with maladaptive shift in the trade-off between survival and fecundity, and that some improvement in some fitness component following climate change might actually signal a maladaptive response reflecting slow evolution and not a change in the direction of selection, but that feedbacks between demography and evolution can result in complex adaptive dynamics. One example of that is that we found instances of tipping points where if you start to accumulate enough maladaptation on one life history component, selection on this life history component becomes weaker and not stronger. And so this actually means that in those circumstances, evolution is slowing down as climate change is accelerating. So you can find situations where you increase the speed of climate change and the population keep tracks perfectly well with the environment, and suddenly beyond some value of the speed of climate change, you will have a large collapse that you could not predict in advance. And those are situations where actually maladaptation is begets more maladaptation. And this existence of this tipping point in adaptation to climate change is something that we are trying to study with colleagues, Matthew Osmond and Sally Otto. And what we are trying to establish is that beyond this example, the simple example of this two-stage model, this kind of behavior is expected in any structured population, be it structured by size, age, or space, as a property of those demographic heterogeneities within populations. So again, I won't have time, I think, to talk to you about tracking climate niche through space, because I'm running a bit short of time. So maybe just to be fast, there are two ways that are a bit a caricature of how you could respond to climate change in species with a broad distribution as trees with a lot of local adaptation within the range. Because climate change is creating local maladaptation, you could respond to that either by not changing your geographic distribution, but evolving different traits to retain the sites where you're already in. So changing your niche limits, but not your distribution. Or you could track the range of conditions to which you're adapted through space by migrating, but keeping the same range of phenotypic distribution within the range. So interestingly, if you have evolution, you could expect any combination of those two processes to occur at the same time or in some proportion. But the early model, the early quantitative genetics model that addressed that question predicted that actually only the second option was likely. That even though there was genetic variation within population and a lot of local adaptation, what would happen most often is that you've got this shifting niche through space, but the same niche limits. So basically saying that all envelope models should be right, even if you explicitly model evolution within the species range. But recent theoretical work shows that those predictions are not completely robust. So if you change things on assumption in this model, you end up with different predictions. For instance, if you let dispersal evolve at the range of the distribution, you predict that actually the distribution is shifting not as fast as climate and with no change in niche limits, but that the distribution will shift actually faster than climate because of the acceleration due to dispersal evolution at the leading edge, and that the niche will shift towards colder climate if the climate is warming, which is very counterintuitive. But because of this expansion dynamics, you predict that actually this whole species is becoming more adapted to cooler climate than in a constant climate. So what we did with colleagues is try to incorporate in those models pollen dispersal, taking into consideration the fact that all those previous models considered only dispersal through seeds or by animals, and that distance of pollen dispersal in trees and plants are often orders of magnitude higher than seed dispersal distance, and pollen seed dispersal have very different consequences on the demographic and genetic dynamics of the population. And so this was a project that we did with Robin Aguilé, Gaël Raoul, and François Rousset. And what we found is that with pollen dispersal, we predict that distribution shifts slower than climate, not faster, and that niche shifts occur but towards warmer climate. So you have species that are not moving up toward cooler climate as fast as you would predict, but they adapt to a warmer climate. And we also found that actually this contribution to pollen to dispersal was actually increasing for a large range of conditions the critical speed of climate change beyond which you predict the extinction of the population. So long distance pollen dispersal would allow persistence under faster climate change. So the reason for that are a bit difficult to understand because pollen dispersal is actually worsening maladaptation at the margin and improving adaptation at the core that we have this prediction in this model. And pollen is slowing down the spatial range shift, which is compensated by climate niche shift. But interestingly, all those combinations are not equivalent in terms of species persistence, and moving slower through space but adapting through phenotypic change is actually allowing you to persist under some faster range of climate change. So to conclude, integrating niche evolution with life history theory helps to reconsider some of our intuition about climate change. It can help parameterizing evolutionary models to be closer to the biology of species and the ecophysiological constraints. It's necessary to plan ahead for climate change, but it also adds to the uncertainties about climate change. For instance, this whole idea of tipping points in the rate of climate change is making prediction really uncertain about when species might persist or crash because of climate, and also it makes the diagnosis of when a species is in trouble more complicated because you need to look at some integrative fitness measure to really conclude about that. So I want to thank you for your attention and patience, and I'm ready for questions.
Thank you, Ophélie, for this really intriguing presentation. And I must say I was caught by quite a number of counterintuitive statements from your side. So I'm sure this has raised a lot of questions that you can now put into the chat, or you can raise your hands if you would like to ask something, and then unmute yourself to ask the question. Anyone ready for starting the discussion? Please raise your hand in the reactions field at the bottom of your Zoom window. You can raise your hand. Benjamin, please.
Thank you. Thanks for your talk. I have a basic question about the first part, the phenotypic plasticity. So I was wondering during your presentation how adaptive phenotypic plasticity is encoded into the genome. Because yeah, this is often a discussion we have with our colleagues, and I would like to have your opinion about it. And the second question is for the second part, did you incorporate the generation time of species when modeling the adaptational lag in your models?
Okay, so for the first question, so is your question, so there are, on the modeling part, we do not model explicitly any genetic or genomic in this exercise. In the modeling exercise, we just change a parameter in the reaction norm to temperature and look at the phenotypic effect. So when we decided about which parameter could be genetically variable in this reaction norms, we indeed had a lot of interesting discussion, but unfortunately, because I wanted to initially find which were the parameters in those complicated model functions that describe phenological response to temperature that were genetically variable, and there's not much data about that. So what we have sometime, and I think Antoine did that on the oak population along the Pyrenees, is we assume a linear reaction norm with a slope and an intercept, and you can measure quantitative genetic parameters on the slope and the intercept and look at what is genetically variable. So in their case, I think what they found is, well, and also the genetic variation might be within or between populations. So you can look at that and say whether it's just the intercept that will evolve or the slope is fixed in the species and will not evolve. But I guess this linear reaction norm is also some simplification of the real reaction norm to temperature, which at least the way it is described in PhenoPhit is much more complex, with sum of degree days and different functions that are abstractions, but it's hard to understand which molecular mechanism or type of genetic variation will affect the parameters of those phenological models. But I think it would be interesting to look at it. What it seems, at least in the oak trees, is that the slope is less variable than the intercept, at least between populations. So this would suggest that, yeah, that the response to temperature is kind of less variable than the ability to be in general earlier or in general later. But I don't know exactly what it means in terms of mechanism of, sensitivity to temperature and development of the organs and stuff like that. For the second question about generation time, we do not, what do you say? How do you answer that? So the model is really a stage structured model with different stages. So the life cycle is described so that individuals do not become adults instantaneously. They wait in the juvenile stage for a number of years, potentially, depending on the rate of transition and rate of growth. So it's not, we do not have a parameter which is called generation time, but the fact that generation time is changing with the parameter of the life cycle is included in there. However, there are different versions of the model, and the simplest version that allows to have nice mathematical results and analytical results assume that the demography changes very fast, so that you are at the stable stage structure almost immediately, and it does not describe this kind of lag that you would also expect in species with very long generation time, in that the structure of the population will not be in equilibrium with the current condition. But we have version of the model that do that, but it's not the one we have explored the most. So this is not the main reason for the pattern that we see in the result I presented to you. But that's an interesting question in itself that I really wish to explore in future years.
Thank you.
Any further questions right here from the audience? I don't see anything popping up. Yes,
Sally has a question.
Sally is up and running, huh? Hi, Sally.
Hi, Sally.
Hi, Ophélie. Great to see everyone. It's rather early here, so I'm not thinking very clearly, but a few things really had me thinking during your seminar, and one is a question that I know you and I have discussed, but the response of bud break to a warming climate, it seems like this would be different for evergreen versus deciduous species. And of course, you had modeled Pinus sylvestris along with the oak and the beech, and I think you've thought about those things. But so that's one question I had, and then maybe tagging on with that it really surprised me, the results early on still surprises me with the Fagus, where you don't have a shift in that bud break timing happening with warming. You just have a relaxation of selection, but the day of year is staying constant in a relatively constant and warming climate. And that seems so counterintuitive to me that I still don't understand that result. Anyways, a couple of things to think about.
So for the deciduous and evergreen, we indeed very clearly see that for the elevation gradient, the fitness landscape for fir are very different from the other ones, and they actually, bud per state is not making a big difference in terms of fitness, and there's a very wide range of dates where it's fine to open your buds. And so we definitely see that when we compare fir to oak and beech along the elevation gradient. And for Scots pines, what we see is something different, which is this, and I think it's not related to so much to evergreen and deciduous. It's more like this requirement for dormancy break by cold temperature, which might affect other species, and I think affects Scots pines because of their more, because they're adapted to cooler climate basically than the other two species. And I think this is something that might affect every species at some point. And this part of the phenological response due to dormancy breakage and cool temperature is not well known and not well parameterized, because we're not typically in conditions where this would happen. But this is happening more and more, actually, now that climate warming is more intense. So I think we might get better data to understand that and how common it is. Concerning the Fagus thing, so it's not that we predict that the dates are not going to change with climate warming. The plastic data are going to change. But what we see mostly with our simulations in this, also in this example and in this gradient, is that you see basically this, something similar to what you see when you go along the elevation gradient, which is that at low elevation, the fitness landscape is very wide, because you can actually open your buds early or late And I mean early because there's not so much frost, and late because you will have enough time to mature your fruit because it's warm enough to do that fast. And as you move up, you have really a small window to do it. And so what we see in this exercise mostly is really that it's this width that is changing much more than the position of the optimum. It doesn't mean that it doesn't change at all. And so when you warm the climate, what's happening is that you're widening those landscapes because, again, you're relaxing the constraint, both on frost and on time to mature your fruits. And so that's the main thing we see. And this is not something that, at least in very theoretical model, you incorporate in general. But it might be very different if we were in a more drought-driven environment, where I suspect that the constraints on phenology will be much stronger if drought was the issue. Because here, I think the main constraint is temperature. It's not precipitation, in our simulation, at least. So I think that's why it explains this pattern. So temperature in itself is not such an issue for plants. It's, drought.
So if you added competition, if not with drought, but if you added competition to the model in terms of growth competition, then it might you might see less relaxation as higher elevation environments become more competitive.
Yeah,
in terms of growth with a longer growing season, right?
I think also in this version of the model, the carbon budget of trees and their growth is a bit very simple. And so I think Isabelle now is repeating the simulations with an updated version where the growth part and the carbon storage part is much more, is described in much better and refined way. And it would be interesting to see if we obtain different conclusions about that. I see that Fred Guillaume has a question.
Fred Guillaume, exactly.
Yes. Hey. Hi, Ophélie.
Hi, Fred.
I have a question about phenophytes precisely. So if I understood correctly, you said that the mechanistic model has been calibrated from experimental data. That's right. And so how confident you are about the predictions when you are predicting fitness or responses outside of this of the set of conditions on which the model has been trained?
So there are different aspects of that. So the model initially was not meant to do that. It was meant to predict species ranges. And so the validation of the model was done previously by confronting the actual distribution of trees with the predicted distribution of this predictive success across different environmental gradients at the continental scale and for many different species in North America and in Europe. So that's, part of the validation is that at least on large scales, it does predict, it does predict fitness sufficiently well so that it does predict occurrence pretty well, and it does predict where the species stops and where it actually can grow. So that's one aspect, and that's why we started from this model initially, as to do that. Then there's a different question: is that are those predictions good to predict a more, local or refined variation of fitness with traits, which might be not, might not be the case, even though you predict at a very global scale pretty well what's happening. And so we tried to try to see if the prediction we obtained with this model along the elevation gradient made sense compared to data on this elevation gradient. That's why also we wanted to focus on some gradient where there was a lot of data. And we did a number of comparisons. So, well, first of all, I find it actually surprising and interesting that the optimal value for bud burst that we predict with the model is very close to what trees are doing, at least in 2 species out of the 3 we're looking at. And this is not something that is built in the model. There's nothing in the model that will force the model to predict that this is optimal bud burst just because the trees do that. There's nothing in the model that would circularly do that. So the fact that we are actually predicting that what trees do is pretty much what they should be doing, at least in 2 species, to me is kind of reassuring. So of course, for oak trees, it doesn't work. So it either means that the model is not working for oak trees or something else is going on there. And I think the 2 are possible. I have an argument for both. So that's another one. And then there are also trends, I mean, more qualitative trends that we try to look at. And the fact that, for instance, reproduction is declining with elevation is very consistent with what we see in the data. The fact that we have signal for, so we also, I didn't show that, but we use a model to predict selection gradients on that of bird burst to see if there was selection for earlier or later bird burst and of varying intensity with elevation. And we did that in the project here and also in some other elevation gradients in EVOLTREE. And this was also kind of consistent, like for instance, the change in selection strength with elevation was globally consistent with what we were predicting. So it's not, you know, quantitative, but at least the patterns were okay. So that's the kind of thing we try to do. So I don't think that you should necessarily take those predictions as exact quantitative predictions yet, but they help you to think about those selection pressures and think about things maybe we have overseen or not considered enough in maybe in theory, but also in the data.
Thanks.
Fred, okay. Is there any other question? I would like to get back to one of the earlier slides where you showed that phenotypic, or the phenotype, the optimum of the phenotype is really linearly related to an environmental measure. And I wondered if there's any argument or in particular empirical support for such a linear correlation. Because we always struggle, or no, not always, but we often struggle explaining why we expect linear relationship between allelic variation and environment. So how is that?
So I would say that, a priori there's no reason to expect the relationship to be linear, and that's why precisely we wanted to do the exercise with Phenotypic to look at, you know, for a given trait and for a given environmental gradient, whether we would have something different. Mm-hmm. And it turns out that actually along the elevation gradients, what we have, so the variation of temperature along the elevation gradient is linear, but this is kind of artificial. It's because we use a linear relationship to extrapolate data from one climatic station along the elevation gradient. So the fact that temperature is varying linearly along the gradient is a bit artificial, but still in our prediction, in this case, we found that the optimal value of the trait was, I mean, the fact that you would assume in this case that it would be a linear function of temperature would work pretty well in this case. But I don't have any special reason to think that it would be general. I don't think this assumption is made because it's more realistic. It's just that it's much easier mathematically to deal with the situation. And there are a number of interesting results. Maybe John, who's here too, I saw, could comment on that. But if the variation, so if the variation was not linear but having some really change in slope in space, for instance, this is a situation where, for instance, you predict that niche limits will stop because it's harder to adapt to an accelerating changing environment when the slope of environmental variation is changing. And so when you consider nonlinear gradients in terms of selection, because this is what is important, and demography, you start to understand a lot of things that do not happen if you have a constant slope through space. But it's still a good starting point. And also, obviously, not such a bad approximation in every case, as our example shows.
Any further questions from anyone?
Antoine?
Antoine is raising his hand. Please unmute yourself. Yeah.
Yeah, okay. I have a question about the adaptational lag in the case where, let's say, you have environmental change and the optimum value is changing. In one of your slides, you show, I think you were referring to Lynch, where you recall that the adaptational lag would remain constant.
Yeah.
Does that consider overlapping of generation? I mean, if you have overlapping of generation, you probably will get some migration load from older population swamping the recent population, and that should actually increase the adaptational lag.
Yeah, but so yeah, so this is happening in the. This is somehow happening in those models that I presented, but it's still. I mean, the value of the lag would be different, but at some point, you will reach a constant lag, where, I mean, if the climate is changing always at the same speed, so you will reach some kind of dynamic equilibrium when the population is adapting as fast as climate. And if it is adapting as fast as climate, the lag cannot change, okay? Because if you're running, as fast as what you're pursuing, you cannot decrease the distance between the two. And so this prediction breaks down when you have, and so the reason why you get to this constant lag is because in a simple model, the strength of selection, which explains the speed of evolution, depends on the distance between the optimum and the mean phenotype. So if you assume a constant genetic variance, for instance, the speed of evolution will be set by this selection gradient, which depends on this distance. So if this distance decreases, the strength of directional selection decreases and you evolve slower. If it increases, the strength of selection of directional selection on the phenotype is increasing and you're evolving faster. And so the only equilibrium is when you evolve exactly as fast as the climate is changing. And this happens given the genetic variance and given the strength of selection for a given distance to the optimum. So this is what happens in this unstructured population model, because you have a direct relationship between maladaptation and the strength of directional selection. When you start to have a structured population, as I showed, but this is true also for other types of structures, this is not necessarily true, because you've got those feedbacks due to demography, which make the strength of selection decline when maladaptation is larger. So then in this case, you don't necessarily reach this constant lag, and you can have a situation where the lag is always increasing until the population goes extinct, basically. Or in some weird case, you can also do that until you lose one of the age or stage in the population. So you can persist, but you have shortened your life cycle. So that's something we've seen as well in some of the cases.
But that should
so
increase.
Indeed, sorry?
That should increase if, let's say, in long-lived species, when you have more structure, more overlapping of generation. Isn't that the case?
So it depends. I wouldn't say that. So it's not always the case that you will reach this case of ever increasing lag in a stage structured population, but in some case you will, for some speed of climate change or for some life history components being affected by climate change. It's not always the case. But it, somehow, I mean, again, this lag at equilibrium, it's interesting to understand, but in some cases, it takes a very, very long time to reach this equilibrium lag. So it's not something instantaneous. So I'm thinking for most population, especially of long-lived trees, we are still in the beginning of seeing the lag increase, and we don't know if it will stabilize, and if we stabilize to a value that will allow population persistence, or if it will stabilize to a value that will cause the population extinction, or whether it will never stabilize because of the phenomenon I was describing. So I think we are at the beginning, of this curve that I've showed, where the lag is increasing, and we don't know whether we'll reach an equilibrium or not. But even with a stage structured population, you could reach an equilibrium. Really, what is important is what is the value of the lag at the equilibrium ultimately, but you might go extinct before that. I mean, the lag might be too large before you reach this equilibrium. You will never reach this constant lag.
Thank you. Thank you.
And I don't see any further hands up at the moment. Please bring up your questions if you still have one. Otherwise, I would say that we think about concluding the seminar today. I appreciate your elaborations on the topic, Ophélie. Thank you very much for having the seminar today. Thank you for everyone listening, discussing, and I would like to hand back to Christian for concluding remarks.
Thank you, Felix, and thank you, Ophélie, very, very much for taking this opportunity and talk about your science. I have just want to say that we make a break now. We will not have a seminar next Wednesday because of conflicting events, but then we will have another one in 2 weeks. And in 2 weeks, we will have Mois Esposito Alonso talking about Arabidopsis thaliana. And we have added a new seminar at the end of it in 3 weeks. Stephen Palumbi will talk about climate adaptation in the ocean. If you haven't registered so far, do it, especially for the last one. There are still a lot of open seats available. And I hope to see you in 2 weeks again. Thank you for coming.